Good afternoon. Myself Dr. Akshay Kumar Vats. I am Assistant Professor in Department of Agriculture, MMDU Mulana. Today I will discuss about hybrid rice seed production technology. First of all, I want to discuss about hybrids. What are hybrids? Any distant cross, any two desirable parents which are distant, when they are crossed, the first filial generation of them is known as hybrid. In case of rice, hybrids produce 20 to 30% more yield than the high yielding varieties. Hybrid seed production should be there. Why? Because it is a profitable venture, it can generate employment and it can give net returns of about 75,000 to 85,000 per hectare. Almost 100 to 105 extra man hours than the conventional method. In comparison to the conventional method of rice production, 100 to 105 extra man hours are needed in case of hybrid seed production. Hybrid rice seed production technology in India, since 1989, we have achieved more than 125 hybrids. More than 125 hybrids are released, and because to develop the hybrids, we need CMS lines, cytoplasmic male sterile lines. We have also the germplasm of more than 100 approximately, more than 100 CMS lines, and we have also developed the restorers. More than 300 private companies are involved in this hybrid seed production, among which the 30 are leading. Government sectors are not competing with the private sectors in case of hybrid seed production in rice. As compared to the private companies, government sectors like NSC, National Seed Corporation, government state seed agencies like in Maharashtra, Karnataka, they are responsible for hybrid seed production just as compared to private seed companies. Most of the hybrid seed is produced by private seed companies. Due to self pollination and strictly self pollination, there is only 0 to 3% outcrossing in rice. Pleistogamy is there. Due to cumbersome and expensive nature of manual emasculation, hybrid seed production is not possible with manual emasculation only. We have to go for male sterile lines. Male sterility system to produce hybrid seed production is really effective in rice. However, there are two types of male sterility system in rice: cytoplasmic male sterility and genetic male sterility. Genetic male sterility is categorized into two types: thermosensitive and photoperiod sensitive genetic male sterility. It is controlled by temperature and the photoperiod, light, exposure to light. In Indian conditions, GMS is not possible. There is no thermosensitive lines, there are no thermosensitive and photoperiod sensitive lines. This system is adopted in China. The system adopted in India is only CMS, and it is really effective. More than 90% of the hybrids are developed through CMS system. GMS system, in GMS system we need only two lines, that is why it is known as two line system. There is A line and R line. The maintenance of A lines is controlled through temperature or photoperiod sensitivity in GMS system, while in case of CMS system, cytoplasmic male sterile system, the cross is done between A and B to maintain A, and A and R to hybrid to make the hybrid. I want to demonstrate in next slide. This is the schematic representation of hybrid rice seed production. See how the A into B crosses give rise to A lines. This is CMS, sterile line first, and the cytoplasm is totally sterile. S denotes for sterile cytoplasm. RR is the fertility restorer gene, and it is also recessive here. So, there is no chances of fertility in this type of cytoplasm. When it is crossed with fertile maintainer line, it gives rise to 100% sterile lines because of sterility in cytoplasm. The sterile cytoplasm dominates here. So, it is the first step to maintain the A line, because you will not get any A line after this. A is a seed parent, you will get the seeds, hybrid seeds on A line, and B is just for maintenance, that is why it is known as maintainer line. So, first line is male sterile line A, second line is maintainer line, that is B. Then this A is further crossed with R. See, capital R and capital R is the nucleus, this is R line, this is a fertile restorer, fertility restorer line. See, the cytoplasm of it is fertile. So, even if we are getting the sterile cytoplasm, the nuclear genes will give fertile lines. So, all the F1s will fertile. So, this F1 will be the hybrid, this is the first filial generation. So, I repeat it, first step is CMS maintenance. The crossed product of CMS with its maintainer line will produce 100% sterile progenies, that is the A line, and maintainer is only for maintenance of A line. When we talk about maintenance of B line, how B line is maintained, how we will multiply the B line, because we need the seed next year also. In subsequent years, we need all the seeds, because hybrid seed production will be there in next season also, and we have to develop the A line, B line and R line. We will need them, we will need the 3 lines. So, CMS system is 3 line system. How will we maintain the B line? B line is maintained through selfing. It is totally fertile line. So, selfing is required there, and by selfing of B line as well as R line, we can maintain R line also through selfing. Now, in next slide, how isolation distance is maintained. This slide is extensively for demonstration. Isolation distance, firstly you will need to know why isolation distance is to be maintained. Isolation distance is to be maintained because we do not need any contamination from the foreign pollens in the field. We need only pollination through the desirable pollen parents. That is why in the hybrid seed production, in the hybrid rice field, we would isolate it through various type of isolations, and we would isolate because in the nearby fields there can be a contaminant. First First is the spatial isolation, that is space, how we maintain the space. See, in the first picture, no other rice crop around the seed production plot is there. In 100 to 500 meter space isolation is there. Nearby fields up to 500 meter there is no rice. And in physical barrier, you can see the figure number 6. See the physical barriers. There are 3 meter strip of dhencha plants, Sesbania. We can sow the dhencha plants around hybrid seed rice field. Also, we can have the 3 meter high polythene barrier to maintain the hybrid seed production purity, to have to remove the contaminants. Number 3 is time isolation. In figure number 5, you can see the time isolation, how we maintain the isolation of time. Time isolation as already means that the crops are sown in different days of sowing. First field in the figure, field 1 and field 2. Field 2 is sown earlier and field 1 is sown late. It is 21 days late, and so as the isolation. When a 21 days late will flower, there will be no flowering in 21 days earlier crop. This ultimately is an example of time isolation, how we maintain the isolation, and there cannot be contamination of foreign pollens. So, these type of isolations according to need in the hybrid seed production we can maintain. Any of the isolation can be maintained according to your needs, whatever possible and whatever is needed in your hybrid seed production according to location and time available. So, next is, these are the approaches when we can synchronize the plants. We need to synchronize the plants for effective pollination in hybrid seed production. How can we adjust the flowering time of plants? Yes, we can adjust the flowering time of plants chemically as well as applying various approaches, like whenever we broadcast urea or urea spray is there in 50 to 60 kilogram per hectare, that is 1 to 2%. Before the third stage of application, the days can be delayed up to 4 to 5 days. We can delay flowering up to 4 to 5 days by using this practice. Also, we can delay the flowering with paclobutrazole spray. The doses are different for male and female lines. Female lines are A parent, A lines. These will be the more, these will be the seed parent. Male lines are R lines, the pollen parents. So, paclobutrazole spray at the dose of 1 kg per hectare, obviously 1.5 kg per hectare because female lines are more in number, and it will require more quantity, chemical quantity. Before the third stage, it is applied and it can adjust date, it can delay date of flowering up to 6 to 8 days. It is the most effective method. Paclobutrazole is a chemical and its spray can delay flowering up to 6 to 8 days, almost a week. If we drain out the water, it is also one of the effective method for male flowering. It is not effective in female flowering. We can't delay the flowering in females. A lines is not affected much. So, draining out water is effective technique It can be applied in any stage, first to fifth, and it can delay 2 to 3 days. The important method which we use, which we apply in the fields in India, is leaf clipping. Leaf clipping is also a effective method. What leaf clipping is? We cut the 2/3 part of leaf, flag leaf. It is a flag leaf which is a barrier in pollination, which is a significant barrier in effective pollination during hybrid seed production. So, when we clip the flag leaf, it is effective in case of male also and female also. It is done in 7th to 8th stage of rice. It can delay up to 2 to 3 days. If we remove panicles, goes on removing panicles at the heading stage, it is also effective. It can delay 5 to 6 days as we choose panicles whichever to remove. In hybrid seed production, we apply GA3, gibberellic acid, but if we delay the application of GA3 according to the stage, it will be effective again in delaying flowering. So, these are the methods of delay in flowering, how we can delay the flowering schedule of the crops. So, when we delay in the application of gibberellic acid, it is effective for male as well as female. At 40 to 50% flowering stage, we can apply it. However, it is applied nearly to heading or before that in normal cases. It can also adjust 1 to 2 days of flowering, it can delay. If we walk, this is also a conventional practice. If we work on the root zone during the panicle initiation stage of rice, it can also delay the flowering. It will delay 2 to 3 days of flowering if we work on the root zone. If we want to advance the flowering, then single superphosphate broadcasting as dose recommended 50 to 60 kilogram per hectare in case of male, 100 kilogram per hectare in case of female. Before the third stage is necessary to advance the flowering. It advances about 4 to 5 days flowering earlier. Muriate of potash. When muriate of potash is broadcasted in the dose of 10 to 20 kg per hectare and 30 to 40 kg per hectare in case of females, before third stage of rice, it can advance 4 to 5 days of flowering. Even when we flood the field with water, it is the effective method in case of advancing the flowering in males. It is not effective in females. It is applied, this method is applied before third stage of the rice. When we apply GA3 at heading stage, gibberellic acid at heading stage, it advances the flowering. The dose is similar in case of male and female, 10 to 15 gram per hectare is the dose. It can advance up to 2 to 3 days of flowering. Boric acid which is a repellent to insects, which is used in the houses. Boric acid can be applied 100 gram per hectare in case of males and 150 gram per hectare in case of females before the third stage to advance it to 1 to 3 days. It can advance 1 to 3 days of flowering. And this is the pattern of sowing the field, how we can go for hybrid seed production in the field. This A line is a seed parent the hybrid seed will be harvested from this line only. R lines on the borders. You can see the blue color lines are R lines. The row ratio is 2:8 here represented. 2 rows of R and 8 rows of females means A line. According to the conditions, we can have 2:6 ratio of hybrid seed production here. If pollination is not effective much, then our line is not effective much, then we can use 2:6 ratio, even 2:4. So, we have to maintain the distance. What are the recommended distance plant to plant and row to row in case of A as well as R? We need to maintain 15 centimeter of row to row distance in case of A lines, and between A and R line we can have 20 centimeters of distance. When we talk about plant to plant distance, it should be 15 centimeter. For effective hybrid seed production, between the R lines there should be 30 centimeter row to row space. I am thankful to NRRI for this effective information. Now, I will focus on application of gibberellic acids. What gibberellic acid can do? It is an efficient and effective plant growth regulator. It can stimulate cell elongation. That is why it is used to enhance panicle exertion in CMS lines. Favorable effects, more favorable effects are there. I can discuss it. It can increase the floret offering duration. It can also increase stigma exertion and receptivity of stigma. It can also promote the plant height. It can also influence flowering, and flowering in parental lines can be adjusted according to this, as I already discussed in the previous slides. It can widen the flag leaf area angle. Flag leaf angle, it is widened through this. It can promote exertion and growth rate of secondary and tertiary tillers in rice. So, gibberellic acid is most important in case of hybrid seed production. This is a demonstration of flag leaf clipping. What are the conditions here? What are the precautions to be maintained? Precautions to be taken are: cut half to 2/3 of the flag leaf from the top, and do not use this type of practice which is crossed here. Never cut below the joint. So, you can see here the effective pattern of flag leaf clipping. This is the list of CMS lines developed in NRRI Cuttack. These are the germplasm of India for various hybrid seed production programs. However, Kalanga 1 is cytoplasm, which is most extensively used, and here are the CRMS lines. 32A lines, CRMS 32A lines have given much more hybrids. It is contributing much more. Kalinga 1 is 32A. In case of Kalinga 1, you can see CRMS 32A. It is most extensively used in case of rice hybrid seed production. What are the agribusiness opportunities through this hybrid rice seed production? Hybrid rice seed production is misconcepted, as farmers see it as the opportunity where it is costly. but as this slide clarifies your statement, if we compare all the costs during 2020, 3 million hectare of area was covered through hybrids. So, there is scope of hybrid seed production and there is a much more employment generation is that. If you want to set up a seed business, this much area, that is 3 million hectare, what it requires? It requires the seed requirement at the rate of 15 kg per hectare. The seed requirement is 45,000 tons. What is the cost of seed? See the opportunities here, agri-business opportunities, what business you can set up in this hybrid seed production. What is the cost of seed? Cost of seed, if we take it at the rate of rupees 3.5 lakhs per ton, then it reflects 1575 crores, rupees 1575 crores. So, scope of hybrid rice in the country, it is 20 million hectare area. It is extendable business, it can be expanded. What is the seed requirement for 20 million hectare? 3 lakh tons. How it is increased from 45,000 tons to 3 lakh tons? And what is the cost at the rate of 3.5 lakh per ton? It will be 10,500 crores. See the extensive opportunities of agribusiness in hybrid seed production of rice. The area required for seed production of 3 million hectare is 1.5 ton per hectare producibility. At the rate of 1.5 ton per hectare producibility is 30,000 hectare. So, whenever you give land for hybrid seed production to any agency, it gives you money. It is a source of money. So, agribusiness opportunities are much more vast. And when we speak about employment opportunity, as I already told, in comparison to conventional one, it needs 100 to 105 additional man days, which is 2 crores man days for 3.5 million 3 million hectare area. Thank you for the source research bulletin. When we cost analysis, when we analyze the cost of hybrid rice, see the comparison with high yielding varieties. Seed cost is 5 kilogram at the rate rupees 50 per kg. Female, when it is 15 kg per hectare, so at the rate of rupees 400 per kg. For hybrid seed, it is 3000, and in case of high yielding varieties, it is there is no male and female separately. So, 250 and 3000 in case of hybrid seed and 2000 in case of high yielding variety. Whenever we talk of labor cost, in case of hybrid seed, it is much more. So, if we give rupees 350 to one labor per day, it comes out to be 250 laborers in case of hybrid seed production and 145 laborers in case of high yielding varieties, conventional method. It comes out to be 50,000, and the fertilizer cost, FIM cost, it comes out to be 5400 in each, because the requirement is equal. Whenever we talk about irrigation, irrigation is also equal to both. So, it comes out to be 30,000. Whenever we talk about gibberellic acid cost, it comes out to be 2000. Other cost, if we goes on increasing the cost, Other cost includes 15,000 if for hybrid seed, and for high yielding variety it is much less. Suppose total cost is 1,05,650 rupees for hybrid seed production, and for high yielding varieties it is 76,400. Average production will be 2 tons per hectare and it will be 4.5 tons. But when it comes about price, the hybrid seeds will be sold at the rate of rupees 90 per kg, and the high yielding varieties will be rupees 20 per kg. The production will give you 1,80,000 of hybrid seed per hectare and 90,000 in case of high yielding varieties. Net income will be much more than the high yielding varieties. So, it is in comparison with high yielding varieties how cost analysis can be done. Thank you for all that. Thank you for the patience. Thank you.